This movie focuses on the first few minutes of embryonic development post-fertilization in an intact C. elegans hermaphrodite gonad. These complex, highly stereotypical events all occur in the absence of new transcription and are completely dependent on the genes expressed in the immature germ cells prior to gamete differentiation. So establishing the right gene expression mechanisms in these cells is essential to successfully kickstarting embryonic development in the next generation. Exploring these mechanisms in this system is what my lab works on. Specifically, we're interested in understanding how global genome organization leads to appropriate gene expression, both temporally and spatially, in germ cells. What are the links between the linear order of genes, chromatin state, and three-dimensional folding of the genome, and how do these properties affect expression of individual genes or groups of related genes? So we study these big questions through the context of two more focused projects. In one project, we're interested in how thousands of small piRNAs are coordinately expressed specifically in the germline. Even though piRNA genes can be individually transcribed, they still cluster in two domains on one chromosome, and they're scattered amid coding genes. So we would like to know whether clustering is important for piRNA expression, and if so, why? We'd also like to know how piRNA genes are selectively activated in these clusters when many of these neighboring coding genes remain silent. And we've identified one factor so far called SNPC-4 that has given us some clues. Now, when we looked at where SNPC-4 binds on the chromosome through ChIP-seq, we were able to find that it binds very strongly to the regions that are covered by piRNAs, and this binding happens primarily in the germline and not in the soma. And if we zoom in, it's clear that the binding is actually distributed across the entire piRNA-rich region, almost coating it. And it isn't restricted to precisely corresponding to individual piRNA loci. And this binding is actually so strong that we can even see it by immunofluorescence as these bright foci in germ nuclei. Now we know that SNPC-4 has a binding partner called PRDE-1, and PRDE-1 also exhibits these same foci. And importantly, each protein is dependent on the other for this special localization. If you disrupt one, then the other no longer forms these foci. Moreover, we can see this by ChIP-seq as well. So in a PRDE-1 mutant, SNPC-4 binding is no longer enriched across piRNA domains. And although I'm not showing it here, each protein, while SNPC-4 and PRDE-1 are both required for the production of piRNA precursors, which puts them very early in the process of piRNA biogenesis. So here's our model so far. SNPC-4 we know is expressed ubiquitously, but PRDE-1 is present only in the germline. So these two factors can only work together to promote piRNA production in that tissue. At this point, we clearly have a link between the organization of piRNA genes into clusters, the coding of SNPC-4 and PRDE-1 across the cluster, and the importance of this for initiating piRNA expression. What we'd like to ask in the future is whether SNPC-4 and PRDE-1 influence the chromatin environment across piRNA clusters, or vice versa, and whether these proteins have any direct effect on the transcriptional machinery at these loci. In a second project, we are interested in understanding how global patterns of histone modifications are established and maintained in germ cells, where autosomes have a fundamentally different chromatin state than the X chromosome. Now, autosomes are primarily enriched for marks associated with active gene expression, in particular H3K36 trimethyl. By contrast, the X is enriched for the silencing mark H3K27 trimethyl. And these two marks tend to be mutually exclusive and are sensitive to each other. And although we know the enzymes that set these marks, MES4 and PRC2 respectively, we know very little about the modifying proteins that help to establish or stabilize these contrasting chromatin states, or proteins that might act as readers or effectors of the downstream processes. Now, in this context, I'd like to introduce UF1. So UF1 is a protein that's present only in undifferentiated cells and is excluded from oocytes, thus its name. Now, UF1 mutants display several subtle germline defects, which I have listed here, none of which are severe enough to disrupt gamete formation or fertility. And then when we performed ChIP analysis of UF1, we found that it mostly binds to germline expressed genes on autosomes and has very little signal on the X, which correlates closely with that chromatin state that I was telling you about. So based on this observation, we then looked at the correlation with these histone modifications, and it was not surprising to find that there was indeed a strong correlation between H3K36 trimethyl levels and UF1 binding. Now, we then asked whether UF1 can affect the levels of H3K36 using immunofluorescence of dissected gonads. We had to use this assay because for reasons that'll become clear in a minute, we couldn't do this, we couldn't do ChIP-seq on all of the genotypes. So looking at immunofluorescence of dissected gonads and looking at H3K36 trimethyl levels, we saw that UF1 mutation on its own does not have a significant effect on the levels of H3K36. Now, when the enzyme that maintains this mark, MES4, is mutated, the level decreases a bit, but is still detectable. However, when both UF1 and MES4 are mutated, the levels of H3K36 trimethyl are much lower than either single mutant. Thus, when MES4 is not around to actively maintain H3K36 trimethyl, a role for UF1 in stabilizing that modification is revealed. So now, as I mentioned before, H3K36 trimethyl and H3K27 trimethyl are considered mutually exclusive marks. So even though UF1 was directly associated with H3K36 trimethyl on active genes, we were curious whether it had any indirect effect on H3K27 trimethyl as a consequence. And so we found by ChIP-seq actually that UF1 does indeed have a substantial effect on H3K27 trimethyls. As you can see, in UF1 mutants marked here in the brown and the red have elevated levels of this mark on the X chromosome as well as on autosomes to a lesser extent. So what is going on at germline expressed genes by UF1 that has such a strong effect on the chromatin state at sites where it isn't even located? So the model we're currently testing is whether UF1 acts as a reader of H3K36 trimethyl. By associating with this mark, UF1 might help to stabilize this modification as a consequence. And we speculate that in the absence of UF1, MES4 has to work even harder to sustain the levels of this mark. And this increased MES4 activity might cause PRC2 to also increase its activity, which would lead to elevated levels of H3K27 trimethyl on the X and on other silenced genes in the genome. And this idea fits with the subtle phenotype we see in UF1 mutants, which might be a consequence of the readout downstream of H3K36 trimethyl. We're following up on these observations and are excited about the idea that we might have found a modifier and or a reader that helps to stabilize the global chromatin state in germ cells and perhaps mediate downstream effector functions. So with that, I'd like to thank everyone in the lab for all their hard work. Deanna pioneered the SNPC-4 piRNA project, which Nancy is following up on. And Cassie initially discovered and characterized UF1, which Maria Teresa is taking over. I'd also like to highlight the hard work of Michelle and Swapna, who are systematically identifying binding sites for all worm transcription factors as part of the MODERN consortium, which I didn't get to discuss today. Thanks for listening.